A memory is created when something is learned, whether by a single exposure or by repetition of the experience. Your sensory organs receive sensory input from the surroundings. This sensory information is remembered very briefly, if only for a few seconds, and then encoded into a bigger store, short-term memory. The process of memory consolidation, which will be the main focus of our presentation today, is the conversion of short-term memory into long-term memory. Within long-term memory, there are even more divisions, and each of these different types are formed in their own way and lead to different kinds of information. But for our purposes, we are only going to worry about one type of long-term memory, declarative memory. Specifically, we wish to focus on episodic declarative memory. Declarative memory is memory for events and for facts, both personal and general. We can consciously access this information, and we're able to communicate it verbally. Episodic memories are memories of what we have experienced personally, and the who, what, where, when, and why that goes along with them. For example, you may have a memory of that vacation you took last summer, and, if asked, you can describe the details with context. You would remember traveling there, who you were with. You can describe the scenery and remember how you relaxed on the beach and how the sun felt on your face. The brain structure to thank for all of these important memories is the hippocampus. The hippocampus is a brain region in the medial temporal lobe. It is part of the limbic system, which is mainly involved in emotion, but the hippocampus is primarily associated with memory and spatial navigation. Functional MRI studies in humans and animals have shown that the hippocampus is important for both encoding and retrieval of memory. By studying lesions of the medial temporal lobe, researchers also discovered the hippocampus isn't involved in all types of memory. After lesions to the hippocampus, short-term memory, non-associative learning, and procedural memory remain intact. Memory that is affected by damage to the hippocampus is long-term explicit memory, like episodic memories of what we experience. A lot of memories that the hippocampus is responsible for are consolidated during sleep. The sleep cycle consists of 5 main stages. Each stage is characterized by specific wave types and certain physiological processes. Long-term potentiation is the cellular basis of memory. It was discovered by Terje Lomo, who was initially observing the response of postsynaptic potentials of cells in the dentate gyres of rabbit hippocampal cells in response to a single stimulus. While conducting these experiments, Lomo then noticed that a high-frequency stimulus followed by single-pulse stimuli on presynaptic neurons elicited prolonged excitatory postsynaptic potentials in postsynaptic neurons. This was the initial discovery of long-term potentiation. The process of long-term potentiation is most studied at the CA3-CA1 synapse involving the Schaffer collateral pathway. Long-term potentiation is a process where the synapse between a presynaptic neuron and a postsynaptic neuron is strengthened as a result of stimulation. Stimulation of the presynaptic neuron propagates a higher frequency of action potentials along the myelinated axon of the neuron. The synapse can be presynaptically mediated through high-frequency stimulation. Now, we'll explore how the hippocampus contributes to memory consolidation. Specific memories are associated with specific synapses. In other words, the memory that you just made about what we have said so far in the presentation is based on a specific set of neurons, presynaptic and postsynaptic. As this memory is recalled more often, the synapse between the set of neurons mediating that memory is strengthened. If that event tied to that memory is experienced again, that synapse will be strengthened further, causing LTP to occur, and the memory will be stored as a long-term memory in the hippocampus. We want to focus specifically on LTP in the hippocampus and how it is important for long-term storage of spatial and declarative memory. So let's first give a brief overview of how the hippocampus fits into the bigger scheme of things. Sensory data from everything you're experiencing every moment, every day is first received by neurons in the cerebral cortex. The hippocampus lies right beneath the cerebral cortex and receives input from structures all over the brain. It is here in the hippocampus that sensory information is integrated and subsequently consolidated through LTP, which was just described. It is now known that the hippocampus is not where memories are stored. It's primarily involved in the initial consolidation of our memories. After consolidation, the information is sent back to its respective brain region for permanent storage. If the memory needs to be retrieved, the hippocampus pulls all of the contextual information back together to produce one cohesive memory. Long-term potentiation in the hippocampus is responsible for consolidating both declarative and spatial memory in the hippocampus. For spatial memory, LTP occurs in a specific subset of neurons called hippocampal place cells. Hippocampal place cells are a pyramidal neuron found in the hippocampus. Each individual place cell corresponds to a specific location in space. As a population of neurons, place cells make up our cognitive map, which is a mental representation of our physical environment. Most experiments conducted to study the intersections of place cell activity, sleep, and memory are conducted in mice. Mice are great models because we can probe the immune, nervous, cardiovascular, and other complex physiological systems that are also prevalent in humans. And in addition, mice are also susceptible to many of the diseases that humans develop, such as cancer and Alzheimer's. First, we'll discuss how hippocampal function is examined with behavioral studies. A mouse can be placed in an enclosure, and place cell activity can be matched with location. This video shows such an experiment. The various colors represent different subsets of place cells. Every time a color pops into the screen, activity is being detected by those neurons. The different place cells fire as the mouse moves around the obstacle course. For example, place cells corresponding to the dark blue color fire when the mouse is in the lower left turn, but those same place cells have no activity when the mouse travels to a different location along the course. Looking at the overall activity in the top left, you can see that each place cell is very localized to a specific location. A similar setup is used in the paper. Explicit memory creation during sleep demonstrates a causal role of place cells in navigation. The experiment demonstrates that hippocampal place cell activity is responsible for creating spatial memories. The researchers questioned if hippocampal place cells during sleep was important. In order to answer this question, researchers looked at place cell activity and the resulting behavior of 5 male mice. To set up the study, a place cell and its corresponding place field had to be identified. This diagram serves the representation of the enclosure the mice were placed in. The mice were allowed to freely roam around the enclosure with various obstacles while action potentials from the place cells were recorded. The white circle inside the blue area outlines the place field of interest. Researchers identified this place field by pinpointing the location the mouse was at in the enclosure when there was neural activity. There were 3 different stages the mice went through in this experiment: pre, sleep pairing, and post. Researchers wanted to test that the mice's spatial memory was better after an experimental condition that involved sleep and hippocampal place cell activity. The pre-session happened before the experimental condition, and at this point nothing had been done to the mice. During the pre-session, the mice were allowed to freely roam around their enclosure. The second stage, sleep pairing, was the experimental condition. For this, the mice needed a reason to remember and seek out this location or place field in the enclosure. In a controlled wake pairing exercise, stimulation of the medial forebrain bundle proved to be rewarding for the mice. This rewarding stimulation was used as the basis of the experimental condition of sleep pairing. During sleep pairing, an artificial association was created between the firing of the identified place cell and the rewarding stimulation. As stated previously, memories replay during sleep, which means the same place cells that were active while the mice was freely exploring are reactivated during sleep. As you can see, there would be a recording electrode in the place cell and a stimulating electrode delivering electrical current placed in the medial forebrain bundle. To create this motivational association, every time activity was detected in the place cell, there would be a resulting rewarding stimulation. After waking, mice participated in the final stage, a post-sleep pairing, where they were once again allowed to freely roam around the enclosure. The results of this study supported the importance of neural activity during sleep and spatial memory. This graph shows how long it took for the mice to find the place field in the enclosure. The white circles correspond to the individual scores of the 5 mice, while the bars represent the averages. Overall, you can see it took about half the time to locate the place field after going through sleep pairing. This proves that the neural reactivation during sleep was crucial to the mice remembering the location. Going back to the model of the enclosure, a mouse's walking pattern after sleep pairing is documented. This mouse went through 4 trials, and each time was placed at a different starting point in the enclosure. Regardless, the mouse was always able to quickly find the place field again. In 3 out of the 4 trials, the mouse immediately moves into the direction of the place field. Occupancy maps from the study show that the mice only show a preference for the place field after they have been asleep with paired rewarding stimulation. The colors range from black, being minimal occupancy rate, to red, which is maximal. Occupancy only becomes intentional after sleep pairing, such that mice have maximal occupancy rate in the place field. This can be further represented graphically. This graph shows no statistical difference between the place field size and the percentage of time spent in the place field pre-session. In other words, the percentage of time spent in the place field corresponds to the percentage of the total area the place field makes up. So, if the place field accounted for 15% of the total area of the enclosure, the mouse spent about 15% of his total time in that area, which is expected if the mouse is randomly roaming about. In comparison, the black bar representing the post-session clearly shows the mice preferred to spend time in the place field being studied. The results of this study proved the mice remembered the rewarding stimulation, even though it was delivered during sleep. More importantly, the mice had to remember the cause of the rewarding stimulation was activation of one specific place cell. After waking up, the mice knew exactly what location they needed to find to trigger that rewarding feeling again. We have just looked at behavioral studies connecting sleep and memory. Another way this topic is studied is with electrophysiology. Experiments on rats have shown that sleep does play an important role in memory consolidation. In the paper REM Sleep-Dependent Bidirectional Regulation of the Hippocampal-Based Emotional Memory in LTP, experimenters investigated how changes in the amount of REM sleep affect memories formed in the hippocampus as well as long-term potentiation. To remind you, REM sleep is a stage in the sleep cycle where the brain and body are energized and dreaming occurs. First, experimenters divided a population of postnatal HP21 male Sprague Dawley rats into three groups. One group of rats, RSD, REM sleep deprived, underwent REM sleep deprivation for 4 hours, which was controlled by surgically exposing the rats' skulls, inserting electrodes to monitor EEG activity on a polygraphic recording device, and waking up the rats by shaking its cage when REM sleep was detected. The same procedure was performed for another group of rats, REM sleep restored rats, which then underwent REM sleep restoration for 2.5 hours. The third group was a control group of rats, REM sleep-controlled rats, which experienced uninterrupted sleep for 4 hours. This group was critically important to understand the data because experimenters measured the LTP response from the postsynaptic cells of the RSC rats as a baseline. Therefore, in order to make any conclusions about the REM sleep-deprived and REM sleep-restored rats, it was absolutely necessary to have a control group to compare to. These rats were euthanized and the hippocampus was surgically extracted. The investigators then performed electrophysiological experiments by inserting a stimulating electrode in the CA3 region and a recording electrode in the CA1 region, as shown in the figure below, while sustaining the hippocampal slice in artificial cerebrospinal fluid. A series of high-frequency stimulus was applied through the stimulating electrode in order to induce LTP. The stimulus acts on the presynaptic cells, activating the synapse by producing current flow. This eventually leads to the field excitatory postsynaptic potentials on the postsynaptic cell, suggesting that the postsynaptic cell will fire more action potentials and for a longer period of time in response to the stimulus, hence the term long-term potentiation. The magnitude of LTP was measured by taking the slope of the field excitatory postsynaptic potentials produced. Field excitatory postsynaptic potentials are a measure of synaptic response produced by a flow of current through activated synapses. The panel B and C illustrate the relative amount of postsynaptic LTP response to a low-frequency stimulus of 10 Hz and a high frequency of stimulus 100 Hz, respectively. This figure supported two main conclusions. One of the conclusions is that LTP amplitude in REM sleep-deprived rats cannot be restored to the healthy LTP level displayed by REM sleep-controlled rats, even if the stimulus intensity is increased to 100 Hz. So even though stimulus intensity was increased, the postsynaptic cells of REM sleep-deprived rats could not fire as much as the postsynaptic cells of the REM sleep-controlled rats. This suggests that REM sleep deprivation plays a role in suppressing the excitatory response of postsynaptic neurons, which ultimately means that the synapses those postsynaptic neurons are involved in are weaker. From before, weaker synapses mean that the memories associated with those synapses are less likely to be able to be consolidated and recalled. The second conclusion that this figure shows is that if REM sleep is restored, there is no increase in LTP amplitude even if stimulus intensity was increased. This suggests that restoration of REM sleep after deprivation may produce a maximal LTP amplitude, which cannot be exceeded even with a larger stimulus. This figure shows that there is a moderate positive correlation between the amount of REM sleep and LTP response triggered in the postsynaptic hippocampal cells. As the duration of REM sleep is increased, the LTP response is increased. The correlation coefficient is 0.61, and this is not high enough to suggest a strong correlation between REM sleep and LTP response. Although it cannot be concluded that increase in magnitude of LTP is directly caused by an increase in REM sleep, the data presented here suggests that there is a reasonable association between the two variables. Behavioral experiments showed that REM sleep-controlled rats and REM sleep-restored rats had better memory consolidation than REM sleep-deprived rats. Ultimately, the results of this paper showed that changes in the amount of REM sleep modulate LTP amplitude and memory consolidation. Ultimately, sleep and memory go hand in hand. Neural processes such as long-term potentiation occur during sleep and improve synaptic connectivity. As detailed in our research, the hippocampus is vital for memory consolidation. This is demonstrated through its role in emotional and spatial memory. It is important to understand the brain structures that are critical to memory formation because it is helpful for developing treatments for sleep-related disorders that affect memory. Understanding the relationship between sleep and memory makes us rethink how our current daily sleep cycle is affecting our memory.